Okay, so see, like I can't even go through my tabs because it's so weird. This is the okay, this is the session we're trying to do. Can you guys see this at least? And why isn't it letting me? Oh my goodness. Okay. I'm gonna quit PowerPoint because it was just working 2 seconds ago, and then hopefully it'll let me open it again. I'm so sorry, guys. Okay, I'm gonna quit it and then see what happens. Okay, that was terrifying. I hope it saved everything. Does this ever happen to any of you guys, or is it just me? Okay, I think it's actually working now. Yay. Okay. Please tell me it saved everything. Yeah, I think it did. Okay, amazing. So let's get started because this is a long session already. Can you guys still see my screen? Because I don't see it red. Actually, no, I think you can still see it. Just let me know in the chat. I have my phone on Teams so that you guys can still send messages and stuff. Okay. Anyways, let's put this in full screen now. I'm assuming you can see it because I feel like someone would have. Okay, everyone's saying that they can. Perfect. Okay, let's get started then. So these were the subjects that were suggested to me at least an hour ago, up to an hour ago, maybe half an hour ago. So we've got haplodiplontic life history, algae in the move to land, gametophyte reduction, taxonomy, so mostly phylogenetic trees, primary and secondary endosymbiosis, these particular life cycles, although I did add a few other reviews. And then someone suggested this, so I put an asterisk here because this was like 4 different lectures, so I don't really know what exactly you want me to talk about. So if when we get there, like I have like a question, like an open-ended one, so that you can drop whatever questions you want to ask about any of these groups or anything like that. Just because I feel like it wouldn't be a good use of time for me to go over like each of these because we already did. But anything like in particular, I would be more than happy to answer. And then also a review on seed plants. Okay, so let's start out with haplodiplontic life histories. Actually, a lot of people asked about this, and so I kind of understand why, because I feel like in first year I was like, ew, what is that? So basically, just as a refresher, in seedless plants, the gametophyte generation is dominant. So the gametophyte generation is the haploid generation. And then in vascular plants, the sporophyte generation is dominant, and the sporophyte generation is diploid. Okay, I'm actually dumb because I switched the rest of this onto later, so I'm gonna go there now. Like I said, literally was working on this forever. Where was that? There we go, haplodiplontic life history again. Okay, so yeah, people asked about the 3 of these, so I'm gonna go over these few pretty quickly, but basically the diplontic life cycle is the kind that animals follow for the most part. So basically what you're doing is you're starting out with your gametes. You have your male gamete and your female gamete, right? So for example, for humans, that would be the sperm and the egg. They are haploid, meaning there is 1 copy of each of the chromosomes. In our case, as humans, we would have 1 copy of each of the 23 chromosomes in either our sperm or our egg cells, depending on whether we are biologically male or female. So the sperm and the egg, or the gametes, are going to fuse together. That's fertilization. You're going to produce a diploid zygote. That zygote is going to undergo some cleavage, and it's going to form the blastula and the gastrula. Again, if you're talking about like humans, for example, then you get mitosis happening. Or actually, that is cleavage. It's just repeated mitosis over and over again. We talked about this on Sunday. And then you end up with your gastrula or whatever, and then it's going to develop further, like the 3 germ layers. So the endoderm, ectoderm, mesoderm, like we talked about on Sunday as well. You're going to get like a whole human baby, and that human is going to grow up, and then that human is going to also produce gametes of its own. So it's going to have its germ cells, which are diploid. They're going to go through meiosis to produce either the sperm or the egg cells, depending on if it's male or female. And then it can also reproduce with another individual of the opposite biological sex, if that makes any sense. So yeah, that's basically the diplontic life cycle. It's diplontic because it's diploid for almost the entire cycle except for the gamete stage. So everything except for the sperm and egg cells are diploid. So the haploid generation is restricted only to the gametes. And then on the opposite end of the scale, you have the haplontic life history or life cycle. So that would be for most fungi and some algae. So basically with this one, what you're doing is you're starting out with your haploid, either like your haploid gametes or whatever, and then so your haploid gametes, okay, they're going to fuse together. That's fertilization. You get your diploid zygote. Instead of that zygote going through mitosis like you did here, when we got that zygote here, it went through mitosis, and then you formed like basically a human after many, many cycles of like cleavage and gastrulation and then like development, right? But here, once the sperm and the egg fuse, or whatever the equivalent of that is for the gametes of fungi and algae, when they fuse after fertilization, you get the zygote, and that zygote undergoes meiosis, right? So again, the difference between mitosis and meiosis is that mitosis produces identical copies of that cell that was going through mitosis. But with meiosis, if a cell goes through meiosis, it produces four cells that are not identical to each other or identical to the parent, and those cells have half of the ploidy that the parent had. So it would produce 4 haploid cells from one diploid cell, right? So we had the diploid zygote is going to produce 4 haploid like cells, and they're all going to be different. Then those can go through mitosis to just basically make copies of those. So that's what you're seeing over here, just making copies of this cell right here, and it's going to continue to go through more mitosis and produce gametes, right? Because it's already haploid, so its gametes are still haploid, because it's just going through mitosis. So if it started out as haploid, it's going to end up being haploid even after mitosis, because again, with mitosis, you're keeping the same ploidy as you had in the parent cell. So that is haplontic. So they're basically opposites, because here the diploid phase is very minor and it's only restricted to the zygote, and then everything else is haploid. So for the diplontic life cycle, the haploid phase is very limited and it's restricted to the gametes, and everything else is diploid, but this is the exact opposite basically. And then for the haplodiplontic life cycle, this is kind of a remix. So this is just a general example, but we did also talk about angiosperms and gymnosperms, which are both types of land plants which follow a haplodiplontic life cycle, and this one is specifically called the alternation of generations. Some algae also follow this life cycle as well, but basically what you're doing is, let's say we start out with the gametes again. You have the male gamete and the female gamete. They're going to fuse together. Obviously, they're haploid, so the fusion of 2 haploid cells gives you a diploid zygote. That zygote is going to undergo mitosis, so it's just going to produce copies, a bunch of identical cells that have the same ploidy, so you can see they're both diploid. And then that cell is going to go through meiosis, so it's going to have its ploidy, meaning it's going to go from diploid to haploid. Then this cell is going to go through mitosis again, so it's going to basically produce a bunch of copies of itself, and then those can go through more mitosis as well, and then you're going to end up with your gametes again, which can fuse and then start all over again. So basically the point of this one is that it's a mix between the two. It's almost 50-50, but it's not, because again, as I said at the beginning, for non-vascular plants, the gametophyte generation or the haploid generation is dominant. And then for vascular plants, the sporophyte generation or the diploid generation is dominant. So it just depends on the specific type of plant you're talking about. But in general, it's way closer to 50-50 than either of these two are. So I hope that makes sense. But when we start talking about specific examples of haplodiplontic life cycles, like when we talk about gymnosperms and angiosperms, that should make even more sense. But yeah, so haplodiplontic refers to a life cycle, don't know why that says life cycle, in which there are multicellular diploid and haploid stages. So again, when we talk about the angiosperms and gymnosperms, you'll see that even the male and female gametophytes, which are haploid, are still multicellular. It's not like for us where our haploid cells, the sperm and the egg, are single cells, right? Like a sperm is one cell and then an egg is one cell. But with the haplodiplontic life history, the gametes that are haploid, they're still multicellular. So you have the multicellular female gametophyte and then the multicellular male gametophyte. So that would be like the antheridium and the archegonium, which we'll look at later. And so, yeah, I literally just wrote this at the beginning as well. But yeah, the haploid generation is called the gametophyte generation, diploid generation called the sporophyte generation. We will come back here with the specific examples in a bit, but I moved these around and then I had to go back anyways because I forgot the first slide. I forgot to move that one. Okay, so let's continue on with the beginning. Okay, so someone also requested this entire lecture. Actually, not just one person, a couple people did. So normally I wouldn't go through a whole lecture again because it's like recorded and I feel like, you know, you could just go back and watch it on like 2x speed if you wanted to. But because so many people had questions about it, I don't want to skip over it. So we're going to do like a very sped up version of this, but if there's anything that I didn't touch on that you want me to touch on, please let me know either through the chat or obviously you're more than welcome to turn on your mic. And then you can also just go back and look at the questions that I had up on D2L and the PowerPoint that I put up in PDF form. Okay, so remember when we talked about how there's a specific group of algae called charophytes, and charophytes are the closest relatives to land plants. So we assume that charophytes were kind of, at some point, a group of charophytes moved to land, and so there were different modifications that were made, like evolutionary modifications that caused the shift from algae to land plants. So it's basically like a speciation event that gave rise to land plants, because originally there weren't, like, land plants came after algae, so we're assuming that there was like a group of algae that went through some changes, like evolutionarily speaking, so natural selection acted on them to make them able to survive and even thrive on land. So that's kind of where we're getting at here. So here's some proof as to why we think that land plants are the closest relatives to charophytes. So first of all, charophytes have proteins that make cellulose. Cellulose is a polymer that is basically the majority of the plant cell wall. We do not have cell walls, so we obviously don't have cellulose. And in fact, we also can't digest cellulose. So for example, like cows and stuff like that, when they eat grass, they can digest cellulose because they have bacteria that are able to digest it. They have the right enzymes. But humans and, you know, a lot of eukaryotes can't actually digest cellulose. So the fact that the plant cell walls contain cellulose and that charophytes have proteins that are able to synthesize it is a big comparison point, because it's not that common. And also the sperm of charophytes, the flagella of the sperm cells in charophytes are very similar in composition to that of land plants. We've talked about this before, but lots of different types of cells have flagella because it's really common for helping with movement and especially directed movement. So away from toxins and towards food sources or nutrient sources or sunlight or oxygen or whatever. And so even though a lot of cells have flagella, depending on the organism that cell belongs to, the composition of the flagella, so the protein composition and stuff like that, is different, but the composition of the flagella found on the sperm cells and charophytes is very similar to the composition of the flagella found on land plant cells. So that's another comparison point. And then also, the microtubules create a cell plate during mitosis. So that is basically the line where cytokinesis is going to happen, or the division of the cytoplasm. So it's that line where you can tell that the cell is about to split into two cells. In animals, it's analogous to the cleavage furrow that we form. But basically, the cell plate that forms during mitosis in charophytes is the same one that forms in land plants during mitosis as well. And so that's another comparison point that leads us to believe this, because we form something different called a cleavage furrow, as I said before. Okay, so how did algae move to land? Obviously, there were a lot of different obstacles that needed to be overcome because of the fact that it's, like, you know, it's pretty clear it's very different living in water and in an aquatic environment as compared to living on land. You're faced with a lot of difficulties with that transition, and so you'd have to develop a lot of different modifications to the structure of the organism in order for them to be able to survive and thrive on land as compared to water. So the first thing was gravity, obviously, which exerts its effects on a much greater scale on land than it does in water. And especially with plants, when they need to grow taller, gravity is a huge barrier to being able to grow tall, because, you know, it's going to try to pull them down, but they need to be able to carry their own weight. So plants have this polymer called lignin, and it's there to stabilize them. So you can find lignin in the roots of plants, and you can also find it on the walls of the xylem to help stabilize it. Because if you remember when we talked about this last time, xylem was found. It's like a tube that's found in vascular plants, and it carries water one in flow in one direction, so it goes from the roots to the shoots. And so because you're having a lot of water and the nutrients dissolved in it flowing really fast, and there's just a lot of it, it's going to rub against and push against the walls of the xylem. So you don't want the xylem to collapse because it's a big part of the structure of the plant. So that's why it's stabilized with lignin, so that it keeps the plant like stable and rigid, so it doesn't get crushed under the weight of all that water and under the force of that water moving upwards. So that is how it kind of defeats gravity in a sense. Another thing would be drying out. Obviously, it's a lot drier on land than it is in water, and they need water in order for them to carry out photosynthesis. Because remember that water is a reactant for photosynthesis. It's part of the light reactions where you go through that water splitting phase. I don't even know if you remember that, but it's not important for this semester. That was a last semester thing anyways. But yeah, so they do need water in order to do photosynthesis. So they need to have access to it, despite the fact that land is a lot drier than an aquatic environment would be. So obviously, water has a tendency to evaporate out of the leaves of the plant or whatever, just naturally through the sunlight and things like that. So what they've done is, well, not what they've done, but what natural selection has done is it has selected for the development of waxy coatings on the cells of plants and on the leaves especially. So this prevents water loss because it's a lot thicker and more difficult. It takes a lot more energy for the water to be evaporated out of the leaves when there's such a thick barrier that's in the way that separates them from being inside of the leaf to outside of the leaf. Like they have to basically go through a really thick waxy coat, and that is really hard to do without expending any energy. So this makes it a lot less energetically favorable, and so evaporation is less likely to happen. So that prevents them from losing too much water just from being on land. Another thing would be dispersal, because in water it's pretty easy for them to disperse their gametes. It's not that hard. So through spores, for example, the currents can carry the gametes and the spores just very easily. They don't really have to worry too much about that. But on land, there's not a whole lot of water that's able to carry them. Although there is some if it's raining, but especially like in drier climates, it's not that accessible. So some plants have developed basically seeds and spores that are easily carried by wind, by rain, so just smaller amounts of water, and some of them even by fire. Some plants have seeds that are fireproof, and actually the fire will break a part of the seed that releases the gamete or like the spore, like it like basically releases the gamete so that it's able to be fertilized much more easily. So sometimes fire actually allows for dispersal rather than just completely killing off the plants and everything in it. And then also, obviously, animals can also help with dispersal, especially when it comes to angiosperms and fruits. I have a diagram of this later as well, but basically, some insects and other things can come and either help with pollination, so it can carry the spores, or if it's a seeded plant, animals can eat the fruit that comes out of it, poop out the seeds somewhere else. Let's say it's a bird, so it eats an apple or something like that, and then it's going to fly to a different place, and then it's going to poop out the seeds because it's usually indigestible, so it has like types of fiber that can't be digested by the bird, for example. So it would just excrete that elsewhere, and then that seed could be planted or like germinated in that other area. So it allows for the dispersal of the gametes without the plant actually having to get up and move, because obviously you can't do that. The fourth thing would be gas exchange. So by that I mean oxygen coming in or oxygen leaving the plant, because that is a product of photosynthesis, and carbon dioxide coming into the plant because that is a reactant for photosynthesis. We need that for the Calvin cycle. So the thing is, whenever plants got that really waxy coating that helped prevent water loss, it also prevented diffusion of molecules, because diffusion is a passive process, meaning there's no energy expenditure, and so it really can only happen over short distances. But we've increased the distance that the molecules would have to diffuse through because of the fact that we added that thick waxy coating. So this is a problem because it makes it a lot more difficult for gas exchange to happen. And so what happened was now, like most plants, have things called stomata, which are basically just pores inside of their leaves, and they can actually control whether the pores are open or closed, and they usually control this depending on whether there is access to sunlight or not, because obviously if there's no access to sunlight, what's the point? Because you can't really go through photosynthesis without sunlight anyway. So they're going to close their stomata to prevent the water from being lost, and then open them up again the next morning, for example, when there's a lot of sunlight, so that they can have the carbon dioxide coming in and then release the oxygen that they produce. The fifth thing is the competition. So even though land has a lot more sunlight than aquatic environments do, or at least access to that sunlight, there's a lot of competition if plants live in close proximity to each other on land, and that makes it kind of pointless. It doesn't, it kind of negates the fact that there was a lot of sunlight there in the first place, because not all plants have access to it. So a lot of land plants have the ability to grow taller in order to avoid competition, and vascular tissue can help them stabilize the structure, like I said before, with the lignin that's found in the xylem tissue, and it also helps them transport water and nutrients throughout. So I want to say, I think it was Sunday, but it might not have been Sunday. But I do have all my slides on D2L anyway, so you can just look at those. But I showed you guys a pretty detailed explanation of what xylem and phloem are. They're basically two kinds of vascular tissue found in vascular plants. Xylem helps to transport water and the nutrients dissolved in it. Phloem helps to transport like food particles. And so basically this vascular tissue allows for these things to be transported throughout the plant, even if the plant grows really tall. Because you can imagine that if it didn't have this vascular tissue, it would be really hard to transport all of the water and nutrients from the roots all the way up to the shoot. So to all the different leaves that are going through photosynthesis and stuff, how are you supposed to transport all the nutrients to there? So that is what xylem and phloem is for, and that's why vascular plants are typically so much taller than non-vascular plants, which are usually very close to the ground. So things like moss, for example, is a non-vascular plant. It's very close to the ground because it doesn't have the vascular tissues to help it transport its nutrients appropriately if it were to grow taller. Okay, so yeah, this is a fern, so this would be a vascular plant. This is a non-vascular plant. This example in particular is a moss, so that's what I was saying before. This fern could grow a lot taller, and it is actually a lot taller than this moss, whereas this is probably the extent of its growth here. So non-vascular plants have some key features. They have the alternation of generations, which I might skip over for now because we're going to talk about that like as its own thing because this was specifically requested. And then also multicellular dependent embryos, again, kind of requested because someone asked about gametophyte reduction, and then apical meristems. So we'll talk about this pretty quickly, but yeah, I guess with alternation of generations, it is an example of a haplodiplontic life cycle. So this was exactly what I was talking about at the beginning when I said that it's kind of like a remix between the diplontic life cycle and the haplontic life cycle, where you have both components happening in almost equal proportions, but there's always one that predominates. And so with the case of non-vascular plants, the gametophyte generation or the haploid generation dominates. And this is actually an example diagram of the alternation of generations type of life cycle in, for example, a moss or another kind of non-vascular plant. So let's go through that pretty quickly. But basically, let's start. I always like to start with fertilization. That's my tip, by the way. Anytime you're faced with a life cycle, this is what I did in first year as well. What I did was I recognized that this isn't exactly like opposites, but it's kind of like you can think of it as opposites for the purpose of understanding the diagrams. That like the difference between fertilization and then meiosis. Like fertilization is where I always start because it's pretty easy to understand. Like no matter what you're talking about some sort of male and some sort of female gamete fusing together, whether that's called a sperm and an egg or something else. Like the point is it's always the male gamete fusing with the female gamete, and then you get a zygote, and then that zygote can become an embryo or whatever, but the point is it's always basically the same. So with the case of non-vascular plants, you have the sperm, which is housed in the male gametophyte. So the male gametophyte is called the antheridium. So the antheridium is the male gametophyte, so it's haploid. However, it is also multicellular, which again brings me back to explaining what the haplodiplontic life cycle is. So I told you guys before that haplodiplontic life cycles involve both a diploid component and a haploid component in almost equal proportions, but both components are multicellular. So this is a multicellular male gametophyte, and this is the multicellular female gametophyte. But the point is that they are multicellular, right? So whereas with us, for example, in our diplontic life cycle, when we're talking about our sperm and egg cells, those are single cells. They are not multicellular. So I hope that also helps you distinguish between the different kinds of sexual life histories. But going back to like non-vascular plants, basically you have the antheridium or the male gametophyte, which houses the sperm. Um, and then you have the female gametophyte called the archegonium, and it's housing the egg. Both of these are haploid gametes, right? So basically, what happens is the sperm needs to kind of swim over to the egg to fertilize it. And it's literally swimming because it needs water to carry it. So you can even see that in the diagram. It cannot be, fertilization can't happen without water. It's required. And that's again because there are no seeds. So dispersal is basically happening just in one way. So yeah, the sperm is going to swim over to the female gametophyte, find an egg, fertilize it, blah, blah, blah. We already talked about this 7 billion times, but when a sperm and an egg fuse, you get a zygote, and that zygote is going to be diploid. Now, the reason why it's diploid is because the sperm is haploid and the egg is haploid, meaning they each have one copy of the chromosome set. So the resulting cell is going to have two copies of the chromosome set, right? So the fertilization process happens in the female gametophyte, the archegonium, right? This big structure right here. And then the embryo also grows inside of the female gametophyte. So it relies on the female gametophyte for protection and for nourishment and things like that. So here's your embryo. Now that embryo is going to mature into a sporophyte. Again, still diploid, but this is the female gametophyte that it was just sitting in, but this is like zoomed out or whatever. And then inside of the sporophyte, they have something called a sporangium, which is basically like a sac. And inside of that sporangium is a bunch of different spores. Now, each of these spores are haploid, right? And so they're going to go through mitosis to produce more haploid spores. And so these spores can grow into the female gameto, or basically like there's like female spores and the male spores. So the female gametophyte spore is going to grow into the female gametophyte. I always say I'm going to change this, and I just keep forgetting. But obviously, the female gametophyte spore matures into the female gametophyte, not the male one. And the male gametophyte spore matures into the male gametophyte. But basically, when that happens, that's where you start back over at fertilization, because now you have the male gametophyte, which matures into the antheridium. That's just like basically the adult male gametophyte. And then you have the female gametophyte, which matures into the archegonium, which is the adult female gametophyte. And again, the archegonium contains the egg, the antheridium contains the sperm. The sperm can swim over using water to swim over to fertilize the egg within the archegonium. You form your zygote, it's housed and nourished and protected by the female gametophyte called the archegonium, and you just basically start all over again. So I hope that made sense, but please, again, I am checking the chat, but just let me know if anyone has any questions at all. I would be happy to repeat anything or explain something in more detail if you'd like. But yeah, so that's basically that. And then one thing that you might have noticed is that the embryo depends on this multicellular structure, which is the female gametophyte called the archegonium. And that's another characteristic of non-vascular plants, is that the embryo requires protection by the female gametophyte. And that is multicellular, which again is just another characteristic of being of following a haplodiplontic life cycle. And so the third thing that we were going to talk about with non-vascular plants is that they have apical meristems. So apical meristems are almost like stem cells, so they allow for lateral growth. Primary growth, meaning this would be like vertical. And so basically you're having these apical meristems located in various areas, but mostly through the shoots and then also in the roots as well. So you're allowing for vertical growth in both places, whether that be like in either case, it's growing taller, but sometimes you need to extend the roots so that maybe you can reach soil that has more nutrients dissolved in there or something like that, or more water or whatever. And then also sometimes you need to grow vertically this way, like in this area, because you need access to more sunlight. For example, let's say there's a lot of competition around this plant and they're all taller than this plant, so it needs to grow taller to be able to access some of the sunlight it needs for photosynthesis. So we would call vertical growth primary growth, meaning growing in length. And you can see that these stem cells, or which we call apical meristems, allow for that primary growth. And it also allows for the production of new leaves and also flowers, basically anything that grows in this vertical direction. I get that the leaves lie down horizontally, but the growth is vertical, if that makes sense. Okay, so now for vascular plants. Oh yeah, I am going to talk about xylem and phloem if you guys want me to. No one actually specifically requested it, but some people like asked me to basically go through this whole session, like the whole like algae moving to land stuff. So let me know before I even get started on this. Would you rather I just skip over this and then talk about something else? Or would you rather I go through xylem and phloem? Does anyone have any suggestions? Okay, perfect. Yeah, so you can tell by the photos, and then I also have a table here. So, I mean, it's pretty clear. So, yeah, I'm actually pretty glad that you guys said that because we have quite a bit to get through. And I'm realizing that even if we take 2 hours, it'll be 9 p.m. Atlantic time. So that sucks for you. So I'm going to try not to go over 2 hours. But basically, some other features of seedless vascular plants would be that their roots are stabilized with that polymer lignin that I was telling you about earlier. Basically, it helps protect the structural integrity of the plant itself, preventing it from collapsing due to like having too much water and stuff like that, just like pressure against the walls of the cells. And they also have really big leaves, which make photosynthesis more efficient because the sun can hit more areas. Like I guess like when the leaf is bigger, the sun is hitting it more, if that makes any sense. Like it's able to capture more of the sunlight's photons. So another thing would be that they have sporophylls on their leaves, which are these parts that you see over here that I circled in blue. And that is where the sporangia that you see, I believe in yellow, that's where they're produced. So the sporangia would be like what we were looking at over here. The sporangia was the sac, and inside of the sac, you have a bunch of tiny little spores, which are all haploid. But the sporangium itself, I believe, is diploid. Because it's part of the sporophyte generation. But basically, yeah, so the sporangia is diploid, and it has the spores inside of it, which can then be dispersed. But basically, in vascular plants, the sporophyte generation dominates, like I said before. But because we're still talking about vascular plants without seeds, it still relies on water for fertilization to happen because the sperm still needs to swim over to the egg. Once we start talking about plants with seeds, it's different because the sperm doesn't need to swim anywhere basically, and there are other mediums that can carry out the process of fertilization aside from water. But just because of the fact that we are still talking about like spores and things, you definitely need water to facilitate that. Okay, so another request, gametophyte reduction. So basically what this refers to is the fact that as we go from talking about seedless non-vascular plants to seedless vascular plants to seeded or seed plants with vascular tissue, you can see this downwards trend in the size of the gametophytes, right? So look at this moss here. This would be an example of a seedless non-vascular plant. This is in light blue in all of these frames. In light blue is the gametophyte generation, right? So what the hell? The gametophyte generation, which I said the name of 5 million times, is called. Oh yeah, it's literally okay. Yeah, it's literally called the gametophyte generation. Oh my God, guys. Anyways, yeah, so the gametophyte generation is a lot larger and more visible than the sporophyte generation, right? So the haploid generation is more visible than the diploid generation in non-vascular seedless plants. But then when we move over to vascular plants but still no seeds, you can see that it's almost 50-50, but that the gametophyte generation or the haploid generation is a lot less prominent, like visually, as compared to the gametophyte generation in the non-vascular plants. And then even more so as we move towards seeded vascular plants, so things like gymnosperms and angiosperms, there is some variation between the two, but in either case, you still have this downwards trend of having smaller gametophyte generations and larger sporophyte generations. So even like when you're looking at, for example, a flower, the part that you see is the flower part, and you don't really see the ovules, which I will show you in a bit when we actually look at specific angiosperm diagrams. But basically, those are the haploid generation, like the ovules and then the pollen. Like you don't really see that as much, but you actually see the literal flower and the stem and things like that. So all of those components are part of the sporophyte generation, and that's the part that we see the most. Same with gymnosperms. When we're talking about like trees, like pine trees and stuff, you would see like the actual pine tree itself more so than you would see the specific seeds and things like that. So that really gives you an idea as to how the gametophyte generation is less and less important. Not to say that it's not important at all, but it's just less big. It takes up less space, and this becomes more dominant. So the sporophyte generation becomes more dominant as you progress from talking about non-vascular plants with no seeds to talking about vascular plants with seeds. And now ready for this? So this diagram truly, like, this is not an exaggeration, like saved my life in first year when I was about to do this midterm, because obviously like the structure of the course was different, so this was a midterm, not like an exam. But anyways, this literally is the most iconic thing ever. Just behold. Okay, so basically it comes from your textbook, but I got it from this website, which is why it's linked here. But it kind of shows you a comparison of the sporophytes and the gametophytes and the roles that they play depending on the types of plants that you're talking about. So this first column is for mosses and other non-vascular plants without seeds, and then this is for ferns and other still seedless, but vascular plants. And then this is for seed plants, so gymnosperms and angiosperms. So when we're talking about the gametophyte generation, it's dominant in the non-vascular plants. It is not dominant in either group of the vascular plants. Whether you're talking about without seeds or with seeds, the gametophyte generation is still reduced. But with the vascular plants that don't have seeds, like ferns, although the gametophyte generation is reduced, it can still live independently and it's still able to perform photosynthesis. So it's not relying on anything, and like it's fine. It's just chilling. Whereas with the seeded plants, so things like gymnosperms and angiosperms, it's really, really reduced, and it actually depends on the sporophyte or the diploid generation in order for it to survive. So it requires the diploid generation for tissue, diploid generation tissue, so things like the like the stem and the flowers and things like that for nutrition and for its survival. So basically what we're looking at here is for mosses and other non-vascular plants, the haploid generation is required, like the sporophyte generation needs the haploid generation. So that's what I was talking about when I said multicellular dependent embryos. Like the embryo needs this female gametophyte for protection, right? And so the embryo is the sporophyte generation, and the female gametophyte is obviously the gametophyte generation. So that's for seedless, non-vascular plants. And then when you get to vascular plants still without seeds, the gametophyte generation doesn't depend on the sporophyte generation. It's just chilling on its own, kind of. Like you see here with this fern. And then when you get to seeded plants, obviously with vascular tissue, the gametophyte generation, the roles are reversed. It now depends on the sporophyte generation. So it's almost the opposite as what was happening with the non-vascular plants. You had, with the non-vascular plants, it was sporophyte depends on gametophyte. But now with the seeded plants, it's gametophyte depends on the sporophyte. That's what I'm trying to say, but I just used 1,000 words to say it. Now let's talk about the sporophyte generation. So in non-vascular plants, it's reduced, and obviously it depends on the gametophyte for nutrition. That's what I said. In seedless vascular plants, it's dominant, but again, it's just chilling. It doesn't depend on anything, and nothing depends on it. And then in seeded plants, it's the same. It's dependent on, but it doesn't depend on the gametophyte generation. So in other words, the gametophyte generation needs the sporophyte generation for nutrition, but the sporophyte generation does not need the gametophyte generation for basically anything. And then these are just some examples where you can actually see, like the sporophyte generation is really small in comparison to the gametophyte generation in mosses and other non-vascular plants. Whereas when we move to vascular plants, but still without seeds, you can see that the sporophyte generation is a lot larger and a lot more visible than the gametophyte generation, which is very close to the ground. So if I was looking at a fern, I would only be able to see this part unless I was really looking at that fern. But like at first glance, it's basically just the sporophyte generation that I see. which is almost the opposite of this. And then in the seeded plants, it's very similar, where basically the only part that you're actually seeing is the sporophyte generation again. So in this case, like you would have the sporophyte generation, you see the stem, you see the leaves, you see the petals and things like that. All of that is part of the sporophyte generation. And the gametophyte generation are these teeny tiny little female gametophytes. So these ovules are contained inside of the female gametophyte, or the eggs, right? And then you have the male gametophytes as the little tiny pollen inside of the anther filaments. So you barely see these, and that's why we would call that also gametophyte reduction. And then very similarly to the gymnosperms, what you're looking at when you look at a gymnosperm, for example, like a pine tree or something, you see the tree, and that tree part, like the bark and like the leaves and things like that, all of that is the sporophyte generation. But then if you really look at that tree, you can see the tiny little cones. You have the female cone, which is the most, like the one that we're probably the most used to seeing. And then you have the male cones. Both of them are very microscopic inside of these cones. You have the female gametophyte is really, really tiny in here, hiding somewhere. And then the male gametophyte is also super tiny in here. But basically, you don't really see these that well unless you're really looking at the plant. So I hope that cleared up gametophyte reduction and also helped you distinguish between which generation depends on the other, if at all, right? Because again, with this, like for the most part, they're just chilling on their own. And I hope it also helped you, I guess, like with understanding what we mean by dominant as well, and that it's the part that you see the most. It's a pretty good rule of thumb to say that the dominant generation is the one that you see. Like with the fern, for example, you see the leaves. You don't really see this tiny little gametophyte generation unless you're really looking for it. Does that clear that up so far? Because I'm not really sure. I don't really see anyone's comments. Okay, someone said sounds good. So if you have any questions, just tell me to go back and I'd be happy to do so. Sorry for the vocal fry. My voice is actually shot. Okay, so let's talk about seed plants now. This was kind of highly requested. So let's talk about basically why seeds are even important, right? Because we were chilling, kind of having spores and things like that, but you know, there are some advantages to having seeds, and that's why they're like favored. And that's why angiosperms, for example, are one of the most diverse groups of plants because their seeds allow them to be super diverse because it allows for optimal dispersal methods. So basically, like I said before, they're very easy to disperse because rather than being forced to be like in order for a non-seeded plant to go through the process of fertilization with its gametes, it's going to need water to physically carry the sperm over to the egg for fertilization. Whereas with vascular plants with seeds, the seeds allow for other types of dispersal methods to happen. So those are the ones that I was listing at first, like even small amounts of water, like rain or fire sometimes, wind, animals eating the seeds and then pooping them out in other places. So all of that helps with dispersal. And also there's like parts of the seed called the seed coat and also the female gametophyte, which I'll show you guys in a bit, but it helps to provide protection from the environment and it also provides nutrients to the growing embryo as well. So again, diagrams are coming. And of course, because it is part of the vascular plant group, the sporophyte generation is dominant. So again, sporophyte generation is diploid. So we're talking about these guys, right? The gymnosperms and the angiosperms. So it still follows that same rule where the sporophyte generation is dominant. Keep this table, by the way. It's super helpful. At least it was for me in first year, just saying. But yeah, okay. So here are the types of seed plants. There's gymnosperms and then there's angiosperms. So gymnosperms usually have their seeds in cones, which is a form of gametophyte reduction, which we said like a million times. But basically, the female gametophyte is now relying on the sporophyte generation, and it's also providing nutrition to the growing embryo. It becomes the food supply because it's reduced down from being its own independent multicellular structure to being basically just food supply. So it's just providing nutrients in order for the embryo to grow. And gymnosperms have no ovaries, and the cones are typically just a bunch of sporophylls that are grouped together to form a cone. And you have two kinds of cones. You have the ovulate cone, which houses the female gametophyte, and then you have the male pollen cone, which houses the male gametophyte. Let me try to get rid of this like software update thingy because it's blocking some of the diagram and it's pissing me off. Update later. Okay, cool. I'll bring back the laser though. Yeah, so basically you have your ovulate cone and your pollen cone. Pollen cone has the male gametophyte. Ovulate cone has the female gametophyte, and then this is your sporophyte generation. But then let's say you're looking at a growing embryo, which is going to be at some point the new sporophyte generation. The female gametophyte becomes used as the food supply for the growing embryo, and then you have basically the integument of the cone becomes the seed coat that also protects the embryo. So yeah, that's a form of gametophyte reduction. And I had this video linked in my other slides, and I'm linking it back in this one again because I'm not going to play it, but please do not underestimate this video. If you want like a really, really good explanation of the life cycle and good detail, watch this video, take notes on it. Like what I would suggest doing is either printing off the diagram and then making, because like you can find this diagram anywhere basically on Google. What I did was I printed it off in first year and then wrote notes basically based on what he was saying. So anything that I didn't already know or like anything that I thought I might forget, I wrote it down like in whatever section of the diagram he was talking about. And then when I went to go study, I like went over the cycle and tried to explain it to myself, which is really lame and weird, but you actually like I highly recommend sitting down and just explaining it to yourself out loud. I know it's really strange, but trust me, it's super effective. Although like because it is open notes, you could basically just write it down and then keep it with you. But anyways, I will also explain it a little bit verbally, but this is going to be a lot more quick than the one in the video, which goes into much more detail. So, you know, pick and choose what you can, depending on how much time you have and stuff, and depending on what Dr. P said the emphasis would be on, right? Make sure that you check in with your prof and see, like, what types of questions she wants to ask about these life cycles, because I wouldn't know because I don't have access to the midterm or the exams or to any of the questions that she's going to ask you. Okay. So, oh my God. Thank you, Queen, for putting in the link. That's actually iconic. Okay, so basically, where should we start? Yes, fertilization. That's my rule of thumb, by the way. I will die on this hill. I always start at fertilization because it just helps me with orienting myself. So basically, here's the male gametophyte. No, here's the male gametophyte called the antheridium. And it basically comes out from the pollen tube inside of the, okay, so basically after fertilization, you have a pollen tube that grows down to fertilize the egg. So the sperm is included in that pollen, and then the sperm can fertilize the egg inside of the female gametophyte. So that would be the archegonium, right? So you have fertilization happening, egg and sperm fuse. Guess what you form? A zygote. That zygote can develop into an embryo. But the point is that embryo is now diploid because the sperm and the egg were, the sperm and the egg were both haploid. So you get a diploid zygote, which becomes the embryo. And then basically what forms around that embryo is the food supply, which comes from that female gametophyte, like I was showing you right here, the food supply. And also you have the seed coat that forms from the integument of the cone itself. Again, just forming another layer of protection and allowing for better dispersal because the seed coat is kind of aerodynamic, so it's pretty easy to disperse as well. But basically, you have the seeds housed in here, and then it's going to, let's say the seed germinates, right? So it's planted somewhere, it germinates, becomes a little seedling, and then that little seedling is going to become a big boy. It's going to be a mature sporophyte. It's going to be diploid, obviously, because so is the seedling. And then, because the mature sporophyte generation has grown up, so it has all of its little baby gametophyte cone thingies. So here you have the ovulate cone again, and then the pollen cone. Again, the ovulate cone houses the female gametophyte. The pollen cone houses the male gametophyte. And basically this is what it looks like. So if you zoom into this ovulate cone, you can see an ovule, like one individual ovule amongst many, right? But we're just going to focus on one at a time. So each of these ovules are contained in a megasporocyte, which has this megasporangium, and then it has the egg in there, or the ovule in there. And then there's a pollen grain that can also come and fertilize it, coming from the microsporangia. So you have the ovule with the megasporangium, and then you have the pollen cone with the microsporangia. The mega one is the female one, and the micro one is the male one. And then the microsporangium is also diploid, right? So it has inside of it pollen grains. So these pollen grains coming from the microsporangium can come in and fertilize the ovules or eggs that are inside of the megasporangium, right? So just keeping in mind that mega, whenever you see mega, for the most part, it's talking about the female gametophyte. And then for micro, it's usually talking about the male gametophyte. But in any case, you have the pollen grains, which is the male gamete, fertilizing the female gamete called the ovule. And then obviously you have fertilization happening as well here. And then it's going to go through meiosis. And then after meiosis, you're going to produce your, basically what happens is with these guys, it's a little bit more confusing because some of the megaspores, like one of them dies off and one of them survives, but that's not really that important for you. But then when you do have, like if we're talking about the one that does end up surviving, you have the archegonium again, so that's the female gametophyte. And then this diagram is very hard to distinguish with the colors that they're using. But yeah, so there's the archegonium and the female gametophyte. And then this would be the pollen tube and the sperm nucleus that are like basically fertilizing the egg nucleus, and then you have fertilization happening. Now, the thing is, this would be the haploid generation, and then this would be the this would be the diploid generation, and this would be the haploid generation. So it's still following that alternation of generations type of thing. But the thing about gymnosperms and their life cycle is that it's a little bit of, more advanced than what was happening with the non-vascular plants when they were being fertilized. So when we talked about, for example, the moss life cycle, that was a lot more simple, but the same principles apply. It's just that now you have different features, things like the microsporangia and the megasporangia and the megasporocyte, microsporocyte, that kind of thing. But then the point is, like, you always have to be able to locate your gametes, so the sperm and the egg, and you have to be able to locate the things like the pollen and the ovule. Actually, ovule just means egg, but things like the pollen grains that fertilize the ovule. And then always being able to understand that after meiosis, you're having the ploidy. Having as in HALVING, so dividing it by 2. Because anytime you go through meiosis, you're producing 4 haploid cells that are all different from each other and also different from the parent. So in this case, basically what went through meiosis was after fertilization, after the pollen grain fertilized the ovule, right? We're just talking about one at a time, although this is happening to a bunch of these at the same time because there's a bunch of ovules here, and the female megasporangium. Basically, what you're getting is after, yeah, so you have this fertilization process happening. I'm just trying to look at the diagram because it's kind of bright. Maybe that didn't even help. But anyways, yeah, so you have your diploid like zygote basically that's being produced. So immediately afterwards, it goes through meiosis because that's going to have the ploidy of that. So because that was diploid, you now have these megaspores and things like that are haploid. So all of these names are a little bit confusing because, like, you know, it's hard to distinguish between them, but just understanding that usually whenever you see mega, think the female, whatever, whether you're talking about the gametophyte or the gamete or whatever, and then the micro is usually a defining feature of the male, especially for gymnosperms. It does differ depending on what you're talking about, but for basically the purposes of this course, that's how it is. So yeah, it's just because from what I understand, you guys don't have to memorize the whole life cycle, but you just need to be able to understand the general idea, and you need to be able to distinguish like the female and the male components. That's what I got from your review class, but I'm not entirely sure if you guys, like, let's say one of you guys asked independently and then maybe you got different information. So, you know, just like be on the safe side. I'm begging you to watch these videos. I promise you I watched the same ones in first year. And I personally found that they were really helpful. I even gave them to my friends, and they were like, oh, good thing you sent me that because it really helped. So, because then you can pause and go back and listen to them explain things. But yeah, so that was the gymnosperms. Now for angiosperms, those are your traditional flowers, and they have their seeds enclosed in ovaries. So that's your ovary over here, this structure, and then it's the one in like light green. And then inside of it, you have a bunch of different ovules or eggs. And so again, because of the fact that it's a vascular plant, you know that the sporophyte generation is dominant. And so you see this, the whole, like basically the plant structure. So the stem, the leaves, the petals, things like that, all of that is the sporophyte generation. It's diploid. And then the gametophyte generation is microscopic as well. So you have the microscopic male gametophyte here, which is basically the pollen, and that's housed inside of the anther filaments. And then the female gametophytes are basically the ovules that are housed inside of the ovaries, right? So what else do I have to say about this? Yeah, it's a very diverse group of plants. So there are a bunch of different flowers or angiosperms that are available on Earth, but basically they all have a similar idea in terms of what happens after fertilization. So the ovules, these eggs, right? Once they are fertilized, they become seeds. So let's say you're looking at this plant, and then it's going to become an apple or whatever after fertilization, or what we know as an apple, a fruit. But the ovules after fertilization become seeds, and then the ovary that the ovules were once enclosed in become the fruit part. So you can see that this part in green, after these eggs are fertilized, so after basically a pollinator comes in, deposits some pollen onto the stigma, and then the pollen tube grows down, carrying some of the pollen, fertilizes the egg, and then that's basically fertilization happening. So each of these become their own little embryo kind of thing. The fertilized eggs become the seeds, and then this ovary that they were enclosed in become the fruit. So when you're looking at this apple, these at some point used to be embryo before fertilization, or eggs before fertilization, and then the fruit part used to be the ovary before fertilization. And so this diagram basically shows you a bunch of different kinds of fruits, but they all follow the same general pattern. Believe it or not, tomato is also a fruit, so you can even see the outline here of the tomato like seeds, which again are just fertilized ovules, and then the fruit part, which is just the ovary of the angiosperm. Okay, and then here's the video. It's basically the twin sister for the gametophyte for the gymnosperm video. Oh my gosh. But this one obviously just talks about angiosperms, but let's look at the diagram pretty quickly as well. This one I prefer, in my opinion, but it is a little bit weird because there's like a weird like endosperm that you get that's triploid, so it has this weird phase. Now, I will just say, when I was in first year, I had a question on my midterm that was like, if I remember correctly, it was like what is like the distinguishing part of like the angiosperm life cycle or whatever, and then one of the options was like a triploid endosperm, and I chose that, and I'm pretty sure it was right, like it must have been right. So that's just basically the extent that I had to go into, but again, I'm not sure because it was a different prof, so obviously some of them emphasize different things, but I'm just letting you know that it definitely was one question on my midterm, so it's worth looking at it at least and understanding that it's there. But I'm not super familiar with this stuff because, again, I didn't have to do this. But generally, what I like to do again is start with fertilization. So you have the egg nucleus and then the discharged sperm nucleus. It's discharged from the pollen. So it just has to come out. And so you can see the pollen tube that actually grew down, like I was saying before, after I was telling you that after like a pollinator comes in and then deposits pollen onto the stigma, you have a pollen tube growing downwards so that it can extend over to the egg and then deposit the pollen, and then the sperm can be discharged from the pollen and fertilize the egg, right? So that's what you're seeing there. You're seeing, hello? There it is. Okay, yeah, so you're seeing the pollen tube here, and then you can see the sperm traveling down, being discharged from the pollen, and then going in to fertilize the egg. I don't really think it matters that much what these antipodal cells are. But yeah, so that's fertilization by definition. Obviously, you have your haploid egg, haploid sperm, you get your diploid zygote, right? Same thing. But then you get something called an endosperm that forms, which is basically like a degenerate sperm. it's a little bit weird, but you can kind of see it in this diagram too. Essentially, you have it all happening in the female gametophyte again, as always. So we would call that the embryo sac in this case. And so the seed coat, which is the same idea as what we were talking about with gymnosperms, that comes from like the integument. And then you have the endosperm here, the triploid one. So 3 copies of each of the chromosomes. And then you have the embryo, that was the product of fertilization. So that's just basically what it is it's a result of double fertilization. So that's how you form that endosperm. And then this entire seed is going to germinate or like grow or whatever, or be planted, and then it's going to produce like the mature sporophyte generation, which we would call the flower. And then here you have the anther, and if you zoom in on the anther, you have the microsporangium and then the microsporocytes. And each of those are diploid, but again, the anther is basically the male gametophyte, whereas, so the anther is the filament, and inside of it is pollen. So you can see that here. You have the male gametophyte is contained inside of the pollen grain, and all of that is contained within the anther filament. And then the ovary is the female gametophyte, and it contains each of the ovules, right? And then, yeah, so they're going to go through meiosis to produce their megasporangium, and then, like, that's again for the female. And then the male gametophyte is going to go through meiosis to produce no, this one doesn't go through meiosis, just the female one does. Okay. But basically, yeah, so you've produced that. You've got your ovaries, your ovules contained inside of the ovary. The microspores that contain the male gametophytes of the pollen grains, like inside of the pollen grains, you have that. Then you have the pollinization pollinization process happening where the pollen is deposited onto the stigma of the flower. The pollen tube is going to grow down. The pollen tube contains pollen, which contains sperm, so the sperm can be discharged upon fertilization so that it can penetrate the walls of the egg or the ovule, and then that's basically going to start the cycle all over again. But all of this, like the fertilization process, it extends from the stigma onto the ovary or the female gametophyte. I think I saw a message on here. Oh yes, just the video. Perfect, thank you. So yes, a lot of these are super confusing. I will not lie. Like even now, like I'm a little bit confused. But it's basically just the general idea is the same in all of these. Like there's always a common pattern. And again, I find that starting with fertilization is always a safe bet. Understanding the difference between mitosis and meiosis in terms of the products that you get from them. So again, just to reiterate, mitosis, you get 2 copies of the same thing you already had in the first place. Meiosis, you get 4 cells, but they're not copies. They're different from each other and different from the parents, and their ploidy is divided in 2. So if you started out with a diploid cell and it goes through meiosis, you're going to produce 4 haploid cells. If you started out with a tetraploid cell, then you're going to produce 4 diploid cells. So you're basically just having the ploidy. But yeah, so the specificities are not in, like, from what I've gathered from your review lecture, like in class, the specificities don't seem too important. However, I don't think that it would do much harm if you watch the videos and then just like at least understood them, because that way you can like, just at least sleep well knowing that you could answer a question if it asked about, for example, the microsporangium or the microsporocyte. Because again, like if I knew what was on your midterm, I would tell you, but that's kind of why I'm not allowed to know. So just be on the safe side. I'm begging you again to watch those videos if you are confused about this at all, or if you're more of a visual person, if you're more of like a reader, you can also find these things linked in your textbook. This is actually taken directly from your textbook, if I'm not mistaken, but I prefer watching videos, so that's why I have them linked. Someone else asked about interpreting phylogenetic trees. So basically, we're just going to go through some of the anatomy of them because the specificities obviously are going to vary depending on what the phylogenetic tree is talking about in the first place. If we're talking about the tree of life, we know that already has only 3 domains, whereas some of them can get a lot more complex if you're going down to the species level. But in general, the tips are going to be the species that you're talking about. So species A through E, obviously these are hypothetical. The branches are going to show you these different like basically the branch point shows you a speciation event, right? So after this, you're forming two new species, and this would be where the common ancestor was. Like what this is saying is that you've now formed from here, you've formed two separate species. So you've had a speciation event, and they both came from the same ancestor. So we would call these two sister taxa, and we would call these two sister taxa as well. And also you can call these, instead of species of interest, you can call them taxa, because taxa is more general. Species literally means species. Like, so you can't use the word species for everything. Whereas with taxa, it basically just means anything in this placement, right? Taxa just means group of. So it doesn't even necessarily, it's not like as restrictive as the term species would be. And then, so yeah, the branch point indicates a speciation event. And then going from left to right, you can see that you're talking about extinct species. And then going to the right, you see that you're talking about present day species, or ones that continue to exist. Because, for example, after the speciation event, if we're looking over here, this ancestor is no longer a thing. It's extinct. Instead, you've had this divergence that's happening, and you've formed a bunch of new species, but this original ancestor doesn't exist anymore. So that's why you're looking at these ancestors versus the present day species as you move from left to right. And then I have a note here that in your notes, like in your lecture, your prof calls the branch points nodes, but both are like acceptable. It's just that, like, if it is multiple choice, just be weary of the fact that these are basically synonyms. So branch point or node, whatever you're talking about, basically indicates that there was a speciation event that took place at that point. And then all of these are showing you the same thing. They're just different ways of drawing it, because you're not looking at the shape of it, and you're not looking at, the orientation. You're looking at the connections that link together the different species, right? So whether it's spun around this way or this way, it's showing you the same evolutionary relationships. Or if it's written, or if it's drawn out with like a more triangular shape, it's still the same thing. Just keeping that in mind, because I know, for example, for me, I had a question on either my midterm or my exam in first year, and it showed me a bunch of different phylogenetic trees, and it was like, which one of these is different? Because they all looked different, like at first glance, but three of them were the same, just in different like representations of it, and one of them was just not like it was actually different. Like, not, it didn't just look different, like the relationships indicated in the tree were different as well. So just being able to understand that you're looking at what is the sister taxa to the other species, so basically which two are linked together by a branch point, and also being able to understand like that whether you switch to get, like, let's say you switched this side with this side, but they were both connected here, that would make no difference. Like, you're still looking at the same thing. It doesn't tell you anything new, because you're still grouping A and B as sister taxa coming from this ancestor, and then C and D as sister taxa coming from the same ancestor. Like, that's fine. But let's say you switch C and D and then you switch them with E, obviously that's going to be a different tree. Does that make sense? So just really being able to understand that just because it looks different doesn't mean it's actually different, and you literally have to look through the different taxa to distinguish between the relationships in that tree versus in the other trees that you're given, if you do get a question like that. And then, yeah, this is exactly what I was saying earlier, is that you can rotate these trees however you want, but as long as you're not switching the order of them or switching the relationships as compared to the branch point or the node, you're fine. Like, you're still talking about the same tree. So in this example, they're showing you that if you rotated this, you're still getting the same tree. So in this case, you see that W is the, what's the word? Oh my God. Sorry, I'm literally blanking. I've been staring at my screen virtually all day. Does anyone know what that's called? The outgroup. Oh my goodness. Okay, so W is the outgroup here, and then it's also the outgroup here, right? So far we're on the same page. But what they've done here is that you can see that here there was a speciation event that led to species X, and then species Y and Z, right? So you can see here you have the speciation event, species X, and then species Y and Z. And species Y and Z are still sister taxa in this tree as well. So this is the exact same tree, even though if I were to just look at this for 2 seconds, I would think they're different trees, but they're not. So just making sure that you understand that you're looking at the actual relationships as opposed to the way that it's drawn out. And then another thing that was also on either my midterm or my exam was this question. It was actually like kind of a give me, like it was one of those things where it's like if you were paying attention, then you'd get it. But then if you kind of happen to gloss over it, you probably wouldn't get it. But basically, it's asking, like, it was exactly this. It was like, what would you call this? And I was like, okay, polytomy. So basically, a polytomy is an unresolved lineage. So technically, you can only have two taxa extending from one branch point. You can't have like three of them like you see here. Whenever you do see three, it means it's incomplete, meaning that they don't have enough information to be able to resolve that. They can't tell you which two are more closely related to each other than they are to the third one. So as far as we know, they could very well all be like, we just don't really know which two are like the sister taxa and which one would have to be like a different group. Does that make sense? So whenever you see a polytomy, it's incorrect, it's not supposed to be there, and it just means it's a work in progress or that more information is needed. So just keep that word in mind, polytomy. Okay, so someone else asked about the difference between primary and secondary endosymbiosis. So primary endosymbiosis explains the origin of the mitochondria and the chloroplast. We talked about this, I don't even know when, but it's on D2L, like in my section of the PAL page, which you should all have access to because I've uploaded everything there. But basically, it explains how we got these two organelles. And it was that theory I was telling you about where an ancestral eukaryote engulfed like an ancestral aerobic prokaryote, and then they lived together in harmony and whatever, and they both benefited because the endosymbiont, so the smaller guy, was given protection and shelter, and the host cell, or the bigger guy, that one was given the products of cellular respiration that came from the bacteria that it engulfed. So it got like nutrients and things like that. And then same thing with the chloroplast, right? We had like the host cell, the ancestral eukaryote, it engulfed a cyanobacteria because that is a type of photosynthesizing bacteria. They both benefited from that relationship for the same reasons almost. So the endosymbiont, or the bacteria, the small one, that one benefited because it was given protection and shelter. And the host cell, so the bigger cell, that one was given the products of photosynthesis. So that would be oxygen and sugar and other organic nutrients. So because both of these relationships were favorable, they persisted over several generations. And at some point, the bacteria, whether you're talking about the heterotrophic aerobic bacteria with the case of mitochondria or the cyanobacteria, they became very reliant on the host cell. So they were unable to live outside of it. And eventually they became a permanent part of the host cell, which we would call the organelle. And then it persisted over generations. So that's primary endosymbiosis. We talked about this. We're kind of familiar with it. But secondary endosymbiosis is a little bit different because it explains how the mitochondria and the chloroplast persisted throughout evolutionary history. So this was like a cool one-time thing, but how did it continue? And how did it spread across the different supergroups in domain eukarya? So we know that like within the eukaryotic domain, land plants have chloroplasts, for example, and algae have chloroplasts, right? So lots of different groups have chloroplasts. And then especially when we talk about mitochondria, land plants have mitochondria, animals have mitochondria, like lots of different types of members within these different supergroups in domain eukarya have these organelles. So the question is, did all of them go through primary endosymbiosis and engulf a bacterium? No, that's not actually what happened. You have something called secondary endosymbiosis, where basically an ancestral well, actually, I have diagrams. I think that's better. But all I'm saying is that secondary endosymbiosis explains how these two organelles that I talked about before, how they were able to be spread throughout the supergroups, and how members of these different supergroups are able to go through photosynthesis, even though evolutionarily speaking, they're in different supergroups. So this should make more sense. So basically, here's the difference between primary and secondary endosymbiosis. So this is showing you the example with chloroplasts. So here you have your ancestral eukaryotic cell. you can see it already engulfed an aerobic heterotrophic bacterium, and you already had that permanent configuration thing going. So let's say this has been generations since then, and now this is a permanent organelle, and it's the mitochondria. So that's what the M stands for. This is the mitochondria. This is a nucleus, but you can see that this cell does not have a chloroplast. So what it's going to do is it's going to engulf a type of bacterium called a cyanobacteria. That's what the C is for, and it's green because this type of bacteria can go through photosynthesis. So it engulfs it. They both benefit for the reasons that I mentioned before and the reasons that I put on my slides in D2L, and this eventually becomes the chloroplast, right? So that's cool. We already talked about this. That's primary endosymbiosis. Now, secondary endosymbiosis, this eukaryotic cell that now has the mitochondria and a chloroplast, obviously with the nucleus as well, is going to engulf not a bacterium, but another eukaryotic cell, a smaller one, right? So you can see this eukaryotic cell is engulfing another eukaryotic cell. So you can tell it's eukaryotic because it has a chloroplast and a mitochondria and a nucleus. In general, it has organelles, so it has to be eukaryotic. But because it's engulfing this, it's the same principle where they both end up benefiting. So it is a symbiotic mutualistic relationship. But you can see that the origins of these organelles are different than what were found here. Here you engulfed the bacteria, so it was a eukaryote engulfing a prokaryote, whereas secondary endosymbiosis is a eukaryote engulfing another eukaryote. So that's really the main difference, is it's a eukaryote engulfing a eukaryote, whereas primary endosymbiosis was a eukaryote engulfing a prokaryote. And this is how you have, for example, the ability to perform photosynthesis, how you have that distributed among several different supergroups within domain eukarya, as opposed to being restricted to just one. And that's just because some of the members of the other supergroups were able to engulf cells from different supergroups that had that ability as well. And so that's why these chloroplasts come from different origins. So when you start talking about protists, which you haven't talked about yet, am I talking about, do I mean, yeah, protists. Yeah, we haven't talked about protists yet. But basically, when you start talking about protists and we look at the different groups of protists, you'll see that we're going to talk about that the fact that some of them have chloroplasts that came from secondary endosymbiosis. So that's why I didn't really talk about it before today, because it wasn't like, it doesn't really make as much sense until you actually start talking about protists, when you can actually see examples of organisms that developed as a result of secondary endosymbiosis. But just in general, recognize that a eukaryote eating, if you will, a prokaryote would be primary endosymbiosis, and a eukaryote eating another eukaryote would be secondary endosymbiosis. So I hope that makes sense as well. Again, feel free to stop me if it didn't make any sense. Yeah, I have this written down. These like specific things are not that relevant to you right now because you haven't talked about protists. So you probably don't even know what like the sarclad is and archaeplastida are, but no. Okay, yes, I took that part out. Never mind. Thank God. Okay, so basically, primary endosymbiosis, again, the prokaryote is engulfed by the eukaryote, meaning the eukaryote eats the prokaryote. That's how you got the first photosynthetic eukaryotes, right? So this also gave you the first supergroup of eukaryotes called archaeplastida. The plastid part is because a chloroplast counts as a type of plastid. There are other kinds of plastids, but in general, the evolution of the first plastids came as a result of primary endosymbiosis, which is why this supergroup is called that. So this is one of the supergroups of eukaryotes. You know how we said that within domain eukarya there are 4 of them? So this is one of the 4, and this group came as a result of primary endosymbiosis. Now, secondary endosymbiosis, again, you have your eukaryote that can photosynthesize is engulfed by a eukaryote that cannot photosynthesize, or one that can, but it has a different chloroplast. So like it's basically a difference in the number of membranes around it, because when you're engulfing it, you now basically have 3 membranes, which you would call the nucleomorph. But again, you haven't talked about that yet, so I'm not going to bring it up. But yeah, so secondary endosymbiosis allowed for the chloroplast to be spread throughout the supergroup. So it allowed for photosynthesis as a result of that, because it takes place in the chloroplast, to be spread among the supergroups. So the SAR-clad, this group of eukaryotes, basically came into existence because a lot of these ancestral eukaryotes engulfed some other ancestral eukaryotes that could perform photosynthesis, and then they also developed the ability to perform photosynthesis. So for the most part, it is those that the eukaryotic cells that can't do photosynthesis engulfing the ones that can, but sometimes you can have both of them able to perform photosynthesis, and that's where you get the stuff with the nucleomorph. But again, the reason why I'm not explaining what that is because you didn't talk about it. But yeah, so that's how you spread the ability to perform photosynthesis across the different supergroups in domain eukarya. And I already talked about this. Yeah, I did at the beginning, and this. Okay, so then someone asked about, well, this is their exact question, but I wasn't really sure like exactly what, and I wish I could like have reached out, but because it's anonymous, I have no idea who wrote it. So does anyone have any suggestions or like any particular questions? Because I can easily pull up my slides, and I can address anything that's confusing about one of these. I have the chat open on my phone so I can see what you guys are writing. I don't think anyone's even typing because normally it tells me people are typing too. Okay, well, let me go to the online one, like the computer one, not the app. Okay, I don't actually know if anyone has any questions. So I guess like, I don't know, like if no one has questions, I don't really know what else to talk about, but we could do some of the games, like the quizzes that we had before. I don't know. What do you guys want to, I just answered everything that was sent to me, so I'm not really sure what to do. I don't mind staying longer. It's just I don't want to waste your time. Guys, it's so weird looking at people's icons and you're just like the only one. Okay. Well, how about we end it here if there are no questions? And then, ew, my screen is still being shared. That's embarrassing. And then if you guys have any questions, message me on Teams, and then we can either like meet really quickly or I can like type out an answer for you. Please watch the videos. Like if you do one thing, actually no, that should not be the one thing that you do, but if you felt from Dr. P, like if you spoke to her and you felt like you should probably get to know the gymnosperm, angiosperm life cycles a little bit more, then please watch them, like even 2 times speed, still better than nothing, and take notes on them. And then what else? I don't know. Because the format of your midterm or like your final exam is a lot different than mine was, just because of the fact that we didn't have like an online exam in the middle of the semester, or like we didn't have a final exam in the middle of the semester. I'm actually losing my mind. So the content was a lot different, and we just had like a big final exam, like the standard thing about everything that was covered that semester. So I'm not actually sure like how in depth it's going to be, but I do know that you guys are all prepared. I'm manifesting it. Even if you don't feel prepared, you are prepared. And like if you just like study, it's open notes. Keep all your PDFs open. You guys know Control F, Command F, whatever. But just be prepared, get studying, and remember it's not actually that deep. Like nothing is ever that deep. If you're stressing over it, if you're like shaking at the thought of doing this exam, please don't. Like, I promise it's not that deep. You're in first year. Even if you want to go into medicine, a lot of med schools, they drop your lowest year if you complete a 4-year degree prior to applying. So it's actually not that deep. Like, I promise, do not lose sleep over this. Just study, do your best, take care of yourselves, and everything's going to be fine. And even if you don't do well, it's not that deep. Okay, someone has a question. Yes.
Okay, I feel like this is kind of a dumb question.
No problem.
But if early eukaryotes didn't have organelles like mitochondria, how did they do cell respiration before like primary endosymbiosis?
So if they didn't have any, so basically, that's a really good question. So it's basically kind of like what happened with bacteria. So bacteria to this day and all prokaryotes don't have any membrane-bound organelles. So they don't, by definition, have a chloroplast. So what they actually do is they have this like the same complexes, but they're embedded in the whole plasma membrane as opposed to being inside of the inner mitochondrial membrane. So rather than doing that whole process within one organelle, they're doing it using their plasma membrane, because really all you need is a membrane of any sort in order to embed each of those four complexes and then ATP synthase and stuff. And so you can have your proton gradient. But yeah, they have to use their actual plasma membrane to do that, as opposed to using the little like inner mitochondrial membrane and then having the protons being pumped between the inner and outer membrane. So it's a little bit different, and that's why it's a lot less efficient, which is also why their cells are a lot smaller. It's one of the reasons why their cells are a lot smaller, because to have the nutrients, like the sugars that you produce, sorry, the ATP that you produce, to have it being transported over such a vast distance as opposed to being in an organelle that's already within the cytoplasm, that takes a lot of time. And that's just because if they're moving through diffusion, like via diffusion, it's just going to take a really long time because the distance is a lot greater. So it's less efficient. So that's why the cell itself is smaller. Whereas eukaryotic cells can afford to be a bit larger in general, for many reasons. But one of them is because they have mitochondria, like several of them within the cell. And so they're producing lots of ATP, and they're all pretty close to the areas where you would need that ATP, just because of the nature of having many of them in the cytoplasm. So wherever you would need that energy, it doesn't take as long to transport it as it would if you were a bacterium without mitochondria. Does that make sense?
Yeah, that makes a lot of sense. Thank you.
No worries. Any other questions? This is very intimidating, just like looking at people's icons. Okay, so I guess if no one has questions, like I said before, feel free to reach out and I would be happy to answer any of them at any point. You guys are so cute. Thank you. But yeah, good luck. Take care of yourselves. I promise it's not that deep. Sleep well. Wake up knowing that you're all baddies and everything's going to be okay. The world's not going to crumble because of one exam. It never will. Not even if there were 5 exams, still wouldn't crumble. So yeah, good luck. Sleep well, and I will see you guys Sunday? No, wait, because it's going to be reading week. It's almost reading week. Oh my God. Okay, well, I guess I'll see you guys after reading week, and hopefully we're all refreshed and in good spirits. Okay, bye, guys.